Hey everyone, my name is Jacob Allred, and today I'm going to be explaining a really interesting and powerful method used to investigate DNA-protein interactions. I chose to research more about Biol lab, which is connected to UNC's Department of Biochemistry, and by reading some of their recent work, I discovered a fascinating method they use known as ChIP-seq to explore genetic mechanisms using stem cells. ChIP-seq stands for chromatin immunoprecipitation combined with high-throughput sequencing, which I will break down into simpler terms throughout this video presentation. In order for us to really understand ChIP-seq, I'd like to start with some of the basics. Let's say we are looking at a bunch of mammalian cells, and within cells are nucleus, and within the nucleus is a bunch of chromosomes. Essentially, chromosomes are made out of what we call chromatin. Chromatin is made up of DNA, histones, which are a specific type of protein, and other proteins that aren't quite as important for understanding ChIP-seq. DNA wraps around the histone proteins to package DNA, and this packaging can regulate gene transcription. Depending on how they've been modified, histone can either activate or repress genes. So in summary, chromatin is essentially DNA wrapped around histones. Now, in a cell, all different kinds of proteins can bind to DNA. For example, one might promote transcription of a gene, and another might repress this gene. With this said, there are some proteins with a more unclear function that requires further investigation. This is where ChIP-seq comes in to help us investigate. ChIP-seq helps us explore DNA-protein interactions and helps us understand the exact binding sites to which our protein of interest is bound. ChIP-seq identifies the location in the genome bound by proteins. To better understand this, let's say we wanted to identify all the regions in the genome bound by a specific protein. Our first step in this process would be to use formaldehyde or a similar strong chemical substance to glue all the proteins bound to the DNA together. This includes all of the DNA-bound proteins, including the ones we don't want to focus on, are glued to the DNA. The next step is to generate small DNA fragments, usually around 300 base pair in size, by essentially cutting them up using restriction enzymes. Next is immunoprecipitation with antibodies, where we isolate the protein of our interest using an antibody that binds to this protein, and all other proteins can be washed away. With this step done, the formaldehyde glue can be reversed by warming everything up to unbind the protein from the chromatin, which holds DNA and histones. After this, we can isolate DNA by washing away the proteins and the histones. So far, my example has only focused on one chromosome, but the overall process of gluing proteins to DNA, cutting up DNA, and the other processes that I mentioned are applied to all the chromosomes in a cell. And on a broader scale, it is usually applied to a large pool of a few million cells, which leaves researchers with a lot of DNA fragments from many, many cells. The next step in ChIP-seq involves using the newly acquired DNA fragments to prepare what's known as a sequencing library by adding sequence adapters to both ends of our DNA fragments. Next, the library is PCR amplified, which is a lab technique used to rapidly produce millions of copies of a specific DNA segment. We can then use millions of reads to create what's known as a genome browser track. The publication and work that I reviewed from Biol lab often used gene and chromosome position of mouse genome. The track helps us to visualize the information for the ChIP-seq reads, and we can better identify regions where a lot of reads were mapped and where very few reads were mapped. Researchers then analyze significant peaks on the genome tracks, which are oftentimes indicated by bars. Researchers can then compare peaks for the same protein in different cell types, like in the lungs versus the liver, or if we didn't know the specific DNA sequence that our specific protein bound to, we could get a clearer picture through looking at the peaks and where the protein might have bound. Furthermore, ChIP-seq can be used to try to determine the functional role of a specific protein by looking at where it binds relative to the genes. For example, researchers at Doan Lab may be looking at a genome track and other data and see that a specific protein binds near the start of a gene, so they might analyze further to see that this might regulate the gene in some way. I hope that this video gave you all a good foundation for understanding how ChIP-seq is used to investigate DNA-protein interactions. Genetic mechanisms are very fascinating, and the work at Doan Lab is very intriguing to me, and thank you all for watching.